Alright everyone, it's time we learn about artificial methods of transformation. So why do we need artificial method again? It is because the natural method has lots of drawbacks. We already discussed about those drawbacks in the previous video on the natural method of transformation. If you haven't already watched the last video, you should first watch them because we'll refer to those videos and various concepts in it again and again in this lecture. So, what are the drawbacks of natural method of transformation? The first is that natural method has a very low success rate. So, for example, if you take some bacteria from a petri dish and try to perform transformation and natural method on those bacteria, you never know that which of those bacteria are competent or whether any of those bacteria are competent or not. Another reason is that bacteria don't easily take up extracellular DNA that often. So overall, the success rate of natural method of transformation is very low. The second reason is that we want to control which type of genetic material is taken up by the bacterial cell. For example, in the natural method, the bacteria usually takes up double-stranded linear type of DNA. So as we have already seen in the previous lecture, that the double-stranded linear DNA are usually taken up by the bacterial cell, but in the artificial method we can provide and thus make the bacteria to take up plasmids because these are more stable. Plasmids are circular double-stranded DNA. Number 3 reason for using the artificial method is that we can increase the size of the DNA that is taken up by the bacterial cell by using the artificial method of transformation. So these are the reasons why we want to control the bacterial transformation process in the laboratory setup and make it as efficient as possible. So as I mentioned, we use plasmids for the artificial method of transformation, but these are not just normal plasmids. We specially prepare those plasmids for our need. So how do we do that? First, we take a normal plasmid, and as I have already said, it is a double-stranded DNA, circular in structure. Then we apply a special enzyme named reverse transcriptase. What this enzyme does is that it cleaves the DNA at very particular positions, and many of these reverse transcriptase enzymes create asymmetrical edges or overhangs, also known as sticky ends. So after the reverse transcriptase acts on the plasmid, it will look something like that. It will create asymmetrical edges. These are known as overhangs. Now what we do is that we take a DNA segment that contains the gene of our interest. For example, if this part of the DNA segment contains a gene that we are interested in, for example, the gene that can produce insulin, we apply the same type of reverse transcriptase on that DNA segment too. So the same reverse transcriptase goes here too, and this cleaves the long chain of DNA in very particular locations as before, and this also creates the same type of overhangs. So after the action of the reverse transcriptase, the gene will look like this. Then we isolate the part of the DNA containing the gene, that is this part, and we put it together with the plasmid. We then apply a special enzyme known as ligase. This fuses the plasmid DNA with the DNA containing the gene. So thus we create a special plasmids that acts as a vector. So this is a vector plasmid. Vector means something that carries something else. So in this case, this plasmid, they carry that particular gene with them within the bacterial cell. So why do we use plasmids for this purpose? As mentioned in the previous lecture, covalently open DNA segments are particularly vulnerable to exonuclease activity. Plasmid DNA are covalently closed, so they are less vulnerable to the exonuclease activity present within the bacterial cytoplasm. Also, the plasmid DNA can have origin of replication initiation sites. For example, if this part of the plasmid has a origin of replication initiation, so the replication will start from this part. That means this plasmid can replicate itself within the bacterial cytoplasm. It doesn't need the bacterial chromosome to replicate. That means when the bacteria will divide by asexual reproduction or cell division, it will make copies of the plasmid independently and can put those plasmids within the daughter cells, thus propagating the property within the daughter cell. So if we can create one bacteria with the plasmid of our choice, we can create a colony of bacteria with the same plasmid within them. So that is the importance of origin of replication initiation sites. We can also have a promoter region within the plasmid. So what a promoter region does is it creates a starting point from where transcription begins. Transcription is the first process of gene expression, which means that if we try to synthesize a protein, so for example, the particular gene that we put in the plasmid is present here. This is the gene and the promoter region is present before it. This promoter region can help transcribe this gene and thus produce that particular protein from that gene without the help of the chromosomal DNA. So if you remember from the previous lecture that we had to put the extracellular DNA segment within the bacterial chromosome so that we can utilize the bacterial chromosomal promoter region for the transcription, without which the extracellular DNA would be non-functional. So in this case, we are already providing promoter region by including it within the plasmid. We can also put a special marker gene within the plasmid. So this is the marker gene. And what we usually do, we use antibiotic resistant gene. So why do we use this marker or mostly antibiotic resistance gene in the plasmid of our choice? I shall describe it here. For example, if we have a plasmid with a particular marker gene, for example antibiotic resistant gene in this example, and we try to perform artificial transformation on a colony of bacteria, some of these bacteria will be able to take up the plasmid within them. So only those bacteria will have the marker gene within them. If we try to plate those bacteria in a selective medium containing that particular antibiotic, then only the bacteria with that particular antibiotic resistant gene will be able to grow and form colonies. The other bacteria that couldn't take up that plasmid will not be able to grow colonies within the petri dish. So thus we can differentiate and separate those bacteria that have successfully undergone transformation. So due to all this reason, we find plasmid a very effective vector media for the artificial transformation process. So how do we perform the artificial transformation process? I shall describe the various methods of artificial transformation in the next lecture. So if you found this video helpful, please like this video and also subscribe so that you don't miss any future update. If you have any question regarding this topic or any other topic related to biology, you can ask them in the comment section and we shall try to answer as many of them as possible. Also, if you have any suggestions, put it in the comment section down below. For the links for further reading or references like research paper in this particular topic, visit the description section of the video. So see you in the next video. Thank you for watching.